Today's webinar is part of the Hardwood Biofuels series put out by the Advanced Hardwood Biofuels Northwest Project. We're a USDA National Institute of Food and Agriculture funded grant research project looking to lay the foundation for a poplar-based biofuels and bio-based chemicals industry in the Pacific Northwest. The program is a consortium of university industry partners. The project is led by the University of Washington. Other universities include Washington State University, Oregon State University, University of California, Davis, University of Idaho. We work with Walla Walla Community College and the Agriculture Center of Excellence, and then also our industry partners, Zia Chem and Greenwood Resources. The way we usually run questions for the webinar, if you have questions as we go along, if you could type those into the chat box, and then at the end of the webinar, we'll go back through them and answer your questions for you. Oh, now I have to. Please give me a second as I go back to the beginning of the presentation. There are a number of graduate and postdoctoral researchers on this project. There are also a number of undergraduates. So today we're actually hearing just from a small subset of the many people that are doing research on this project. And we're focusing today on the conversion research that's happening at the University of Washington. I'm here with Chang Dou, Hanna Horhammer, and Oliver Jan. I apologize if I pronounced names wrong. And the idea was to both give them a chance to practice giving a presentation, particularly to audiences who may not necessarily know as much about chemistry, engineering, conversion processes. So at this point, I'm going to hand it over to Chang, who's going to introduce the concept of converting poplars to biofuels.
Thank you, Noel. Good morning, everybody. My name is Chang Dou. I'm currently a PhD candidate at the University of Washington. So as representatives of a large number of graduate students and postdoc fellows in Advanced Hardwood Biofuel Project, it is a great pleasure for us to be here and present our current work through this webinar. And thanks AHB and especially the extension team for providing this opportunity. As mentioned by Noel, I, together with Hannah Oliver, will present our research about how to convert hybrid poplar into biofuels. And I will first make a brief introduction of the project. After that, each of us will take the time and talk about the individual research we are working on here at the UW. Before we start to talk about our work, next slide, we would like to first acknowledge the funding resource and our collaborators. All the research we will present today is part of the AHB project. So in particular, we would like to acknowledge USDA for the funding support. We would like to thank Greenwood Resources and Zia Chem for their long-term support. Our communications brought us new insights and great ideas. And we believe such interaction between academia and industry will facilitate the future biofuel industry in the Pacific Northwest. All right. Let's get started. The main objective of our research is to convert short rotation hybrid poplar into fuels and chemicals. Some of you may have watched previous AHB webinars and know why we choose poplar as feedstock for biofuel production. So in brief, poplar as a woody biomass can be available all year round. It is easy to propagate, cultivate, and it is highly productive. We can also even grow poplar on marginal lands. So different from most previous poplar biofuel research, the trees we studied in our project grow as short rotation poplar corpus. So why short rotation poplar? So let's take a look how poplars grow in short rotation plantation. Initially planted from cuttings, poplars are grown as high density for 2 years to properly establish the root system. The harvest was conducted using agricultural machinery instead of regular logging equipment. This makes the harvest process fast, convenient, and efficient. In the following years, the developed root system will stimulate the growth of next cycle, supporting greater biomass accumulation. That means we can get the biomass every 2 to 3 years from the short rotation coppice plantation instead of waiting for at least 10 years from regular tree farm. As you can see, the visual experience of the harvest poplar is a mixture of branches, leaves, bark, and chips. From that, we remove the leaf material and prepared our biomass, the poplar coppice without leaves. Today, we will focus on the conversion side and talk about how we can make the renewable fuels from that poplar coppice sample. So this diagram shows the conversion process from poplar to drop-in fuel. Poplar is high in sugar content. However, as other plants, it developed a rigid structure like a wall to keep the sugars from being taken away. Within the wall, sugars strongly bind to each other and form into bricks. And the bricks are further glued together with a special cement called lignin. So that special structure is called lignocellulose. It is made from 2 sugar polymers, cellulose, hemicellulose, and the cement lignin. So in order to produce fuels from poplar, one of the key processes is to extract the sugars from the tree. With the help of steam and acid, we could break down the wall and open up its structure. As a result, the bricks become swollen and extensively exposed. We call that process steam explosion pretreatment, as you can see here in the second picture. Then we add enzyme, which are special proteins work as shovel to peel off individual sugars from the sugar brick. That process is called enzymatic hydrolysis. Once we get enough sugars, microorganisms will ferment the sugars into fuels and chemicals. After we get acetic acid or ethanol from fermentation, catalytic reactions would help us to further convert them into drop-in fuels. From 3 aspects, we try to improve the poplar to fuel process. I mainly studied the idea of improving the sugar yield by using different poplars. Hanna studies the impact of ash removal in sugar yield, and Oliver will talk about his research in making jet fuel from ethanol. Here I'm showing you the pretreatment facility at the UW. On the left side, the boiler makes steam for the pretreatment, and in the middle is our steam explosion reactor, where the pretreatment happens. Basically, the reactor chamber is a vessel that holds the biomass at high temperature and high pressure. On the right side, that is the control panel which monitors and controls the entire reaction system. How does the steam pretreatment work? Here, I will briefly go through the process. The biomass, poplar coppice without leaves, are fed into the reactor chamber from the top of the reactor. After that, we close the top valve and open the steam valve and introduce the steam into the biomass for pretreatment. After reaching the specific temperature, pressure for a certain reaction time, we close the steam valve and open the blow valve. A big explosion releases the pretreated biomass and blow it into the collection tank. The pressure release happens in a second and comes with a big bang. Thus, we call our pretreatment process steam explosion. After that, we collect the steam pretreated biomass from the collection tank. As you can see in that bucket, the pretreated biomass looks like a slurry. After steam pretreatment, we do paper filtration and get solid and liquid fractions. So in solid fraction, we mainly have cellulose and lignin. In the liquid fraction, we have hemicellulosic sugars and degradation products. For the solid fraction, we hydrolyze cellulose into glucose using enzyme, and the glucose can then be fermented. And for the liquid fraction, we can directly ferment the hemicellulosic sugars into products like ethanol and acetic acid. After dehydration, we could obtain ethylene from ethanol. With further catalytic reactions, we could convert ethylene into jet fuels through oligomerization and hydrogenation. So Oliver will mostly talk about this process. Again, three of us will talk about our individual projects from different approaches. I, myself, will discuss how we can improve sugar yield by using different poplar hybrids from different sites. Hanna will talk about ash removal impact to the sugar yield. And in the end, Oliver will present his work in convert ethylene into dropping fuels. So here comes the first talk. So as part of my research project, I investigate the sugar yield performance of different poplar hybrids from 2 different sites. In collaboration with Greenwood Resources, we select 2 hybrids. Based on the productivity, they were labeled as low productive and high productive hybrids. Both hybrids are harvested from 2 sites, Jefferson in Oregon and Clarksburg in Northern California. You can see, for example, in Jefferson site, the low productive hybrid had 6 dry tonne per acre yield, while the high productive hybrid had 8 tonne per acre per yield, which is about like 2 dry tonne per acre more than the low productive one. Again, 2 hybrids, low productive and high productive, were collected from both sides. From that, 4 poplar feedstocks were processed using steam pretreatment condition at 195 degrees C with 3% SO2 for 5 minutes. The solid fraction were further hydrolyzed with 5-FPU enzyme loading. So how does the chemical composition of the raw biomass look like? For Jefferson site, no difference was observed for sugar and lignin composition between 2 hybrids. The only difference was found in ash and extractive contents. The low productive hybrid has lower ash but high extractives. Similarly, for the Clarksburg site, the ash and extractive content were different between hybrids. Given the similar sugar content between hybrids, we hypothesize that the sugar yield will be similar within each site. This table shows the chemical composition of the solid fraction. Basically, we didn't see any significant difference between low and high productive hybrids in sugar, lignin, and ash content for both Jefferson and Clarksburg site. However, when we look at the sugar recovered in the liquid fraction, things change. For Jefferson site, more glucose, xylose, and other minor sugars were recovered from low productive hybrid. Overall, for every ton of biomass, the low productive hybrid has about 40 kg more sugar in the liquid fraction. That is about a 40% difference. Similar for Clarksburg site, the low productive hybrid has about 20 kg more sugar recovered in the liquid fraction. This figure shows the hydrolysis yield from the solid fraction. The x-axis lists the two hybrids for both sites. The y-axis represents the polymeric sugar to monomeric sugar conversion in percentage. Basically, from this figure we can see that no significant difference was found between low and high productive hybrids for both sites. Putting all the monomeric sugars in hydrolyzed solid and liquid fraction together, this figure shows the overall sugar yield after pretreatment and enzymatic hydrolysis. Basically, it tells us the total amount of fermentable sugars that can be utilized by microorganisms. As y-axis shows, we calculate the overall sugar yield based on kilogram of sugar per ton of raw biomass. Interestingly, for both sites, we can see the low productive hybrid has higher sugar yield. In Jefferson site, the difference was 19%, and in Clarksburg site, it was 9%. So recall the previous slides, we had a similar raw biomass composition, similar solid fraction composition, and similar hydrolysis yield. The only difference comes from the liquid fraction. Basically, we recovered more sugars in the liquid fraction, resulting in more overall sugar yield from the low productive hybrid. But if we take the popular land productivity, as I showed in the very beginning, into consideration, we may have different results. So here we propose an integrated model that includes both conversion yield and land productivity. In this new system, instead of using conversion yield or land productivity, we have a new standard, land sugar productivity. That is how much sugar can be produced every unit land each year. Because the sugar is a key in biofuel production, and it is affected by both conversion yield and land productivity. As shown in y-axis, we calculate the amount of sugars we can get from 1 acre of land every year for all the popular hybrids. Here we can tell the high productive hybrid is more competitive in land sugar yield in Jefferson site, while both hybrids perform similarly in Clarksburg site. So that number will give us a hint to select the best hybrid and maximize the final revenue per unit land. For example, in Jefferson site, the corresponding annual land sugar yield for high productive hybrid can be 10% higher than the low productive one. In summary, we found out that although different hybrids have similar chemical composition for both sites, they behave differently in conversion. The low productive hybrid had 9 to 19% higher overall sugar yield than the high productive one. More importantly, it is interesting to see the land sugar productivity varies between hybrids and sites. That information will be helpful for feedstock growers like tree farm owners and feedstock users like future biorefineries in selecting their specific hybrids. So now, that is what I have for my talk. I'm going to turn our time to our second speaker, Hanna.
So, hello everyone. My name is Hanna Horhammer, and I joined this research group here at the University of Washington as a postdoc about 1 year ago. Today I'll talk about how to increase sugar yield by removing ash from biomass before steam explosion and enzymatic hydrolysis. Here we have the whole poplar conversion process that Chang already presented to you. In my case, an ash removal step has been added before the actual conversion process. Ash can be a problem when processing biomass. Ash can harm the process equipment, but also decrease the efficiency of the reactions in the process. Therefore, it might be useful to remove ash already before processing the biomass. Certain types of biomass contains more ash than others, and then the ash problem becomes even more relevant. Our poplar biomass without leaves contains about 2% ash. This is a scheme of my research on ash removal. Even though my focus was on de-ashing, I have worked my biomass through this whole process. I started with raw biomass, did de-ashing, SO2 impregnation followed by steam explosion, and ended with enzymatic hydrolysis. The biomass I used was second rotation 2-year-old poplar coppice from the Jefferson site in Oregon. From the plantation we received biomass containing chips, bark, and branches, no leaves. During de-ashing, the biomass was treated with a dilute acid solution at 80 degrees Celsius for 3 hours. This was done in plastic bags in a water bath. The biomass got a somewhat orange-like color after de-ashing, as you can see from the picture on the slide. After de-ashing followed SO2 impregnation, steam explosion, and enzymatic hydrolysis using the same conditions as in Chang's case. I will present results from 2 different cases. First, conversion of original biomass, where no de-ashing was taking place. And then the other case, conversion of deashed biomass, where a de-ashing step was included to the process. Here is the chemical composition of the 2 different biomass samples before steam explosion. So we have untreated original biomass and then de-ashed biomass. The picture above the table shows what these samples look like. The sugar content is very similar, whereas the percentage of lignin is somewhat higher for the de-ashed biomass. When we look at the ash content, we get the confirmation that the de-ashing step really removes a significant part of the ash. The ash content decreases from 2% to 0.6%, which means an ash removal of about 70%. There's about 10% extractives in the original biomass and less than 5% in deashed biomass. We also looked into specific metals. Calcium and potassium are the major metals in poplar, and by deashing, these metals significantly decrease. This table shows the chemical composition of the solid fractions after steam explosion. Again, for the two different cases, original biomass and deashed biomass. As you can see from the picture above the table, we now deal with biomass that has been broken down by steam explosion. The deashed solids have somewhat lower sugar content, which means that we lose some sugars because of the deashing step. The lignin content is about the same in both cases. Again, the ash removal is confirmed when comparing the ash contents of the original solids and the deashed solids. The calcium content is lower for the deashed biomass, and after steam explosion, there are no traces of potassium, magnesium, or phosphor. Now let's take a look at the liquid fraction after steam explosion. The liquid is a brownish liquid, as you can see from the picture. We have more glucose and xylose in the liquid fractions when the biomass was deashed. The minor sugars are on the same level. For the original case, there was in total about 170 kilograms monomeric sugars per tonne, whereas for the deashed case, the corresponding number was about 200. This means that we end up with about 20% more monomeric sugars in the liquid fractions when the biomass has been deashed before steam explosion. The amount of monomeric sugars in the liquid fraction makes a big difference when evaluating the processes with regard to sugar yields. These monomeric sugars can be directly fermented into byproducts. This figure shows the yields after enzymatic hydrolysis for the two cases. On the y-axis, we have the cellulose to glucose and xylan to xylose conversion given as percentage. In other words, how big a part of the sugars have been broken down to monosugars during enzymatic hydrolysis. From the figure, you can see the improvements of the enzymatic hydrolysis when the biomass has been deashed before the process. The green bars show the conversions to glucose. For the original case, the glucose conversion is about 60%, but after deashing, the glucose conversion is close to 80%. This means an increase of about 20% for the deashed case. The purple bars show the conversion of xylose, where the difference is not that significant, only about 5% higher for the deashed case. But in bigger scale, such improvements gain the overall sugar yield for the process. Now let's add up all the monomeric sugars from our process. First, we have the monomers in the liquid fraction after steam explosion. So the sample that looks like this. And then we also have the monomers from the enzymatic hydrolysis, these ones, which earlier came from the solid fraction from steam explosion. Adding the monomeric sugars together, gives us the overall monomeric sugar yields for the processes. The overall monomeric sugar yield is shown in this figure as kilogram per ton biomass. We have the monosugars glucose, xylose, and minor sugars for the original case here. And then in the same way, the monosugars for the deashed case. We can see that the sugar yield after deashing is about 17% higher. This can be explained by that after deashing, we recovered more sugars in the liquid fraction after steam explosion, and also had a better conversion during the enzymatic hydrolysis of the solid fraction. So here I'm summing up the conclusions of my presentation. Ash removal before steam explosion and enzymatic hydrolysis decreases ash content by 70%, increases monomeric sugar content in liquid fraction after steam explosion by 20%, improves the conversion during enzymatic hydrolysis for both glucose and xylose, improves the overall monomeric sugar yield by 17%. So what have we learned this far? Sugar yields can be improved in different ways, either by choosing the right biomass or clone or site as Chang presented, or then by removing ash from the biomass before processing, as you heard in my part of the presentation. Okay, now Oliver will take over.
Hi everyone, my name is Oliver Jan. I'm a fifth-year PhD student at the University of Washington. I've been working on this project for about the last 2.5 years now. And my research as part of this large consortium is focusing on the conversion of ethylene into drop-in fuels. So, from the results presented from Hanna and Chang, the eventual goal of the process is to take lignocellulosic biomass, aka hybrid poplar, and convert that into a chemical known as ethanol, or acetic acid. And with the ethanol, at least, what we can then do is to do a catalytic conversion from the ethanol into drop-in fuels. And the way we would take care of that is through a process where we introduce a special type of material called a catalyst. Now, the overall process flow diagram as to how we would accomplish this goal is basically proportioned into 3 different types of stages. In the first stage, we would take the ethanol and we would dehydrate the ethanol to form ethylene and also residual water as well. The next step would be to oligomerize the ethylene into longer chain hydrocarbons. Now, the thought process behind oligomerization is, rather than taking tens of thousands of these monomeric units and forming polymers, when we make oligomers, we are actually taking more discrete amounts of these types of monomeric units and making shorter molecules. By taking advantage of this process, what we can then do is create more of a liquid product instead of a solid product. The next step in this is to hydrogenate the resulting olefins to produce a jet fuel or diesel type of material. The way we would accomplish this is using hydrogen as a reactant in this process. However, the biggest conundrum in this process flow diagram is figuring out how to oligomerize the ethylene. The dehydration reactor and the hydrogenation reactor are fairly straightforward industrial processes. However, the oligomerization reactor process is quite difficult to accomplish. And that's where the aim of this research comes into play. And our goal is to determine a catalyst to carry out this process. So here at the University of Washington, we've gone ahead and created a oligomerization system where it's basically a packed bed reactor, and in the gas manifold, what we have is our nitrogen gas and our ethylene. We are using pure ethylene gas as our reactant, so we're assuming that we've already gone through the dehydration stage to produce the ethylene. The next process downstream is the reactor itself, which is located in the middle, and this has the schematic shows a packed bed system. So those little beads of catalyst that you see, that is atypical of a packed bed system. And what will happen is that the ethylene will flow from the top of the reactor and through the reactor past the packed bed system. And at the packed bed itself will will the reactions actually occur. Next, the reactions, once they are finished, the products will be continuously swept through the reactor and into a downstream condensing system, where the condenser will be used to collect any liquid product that would be produced. Now, furthermore, the entire system of the entire pressure of the system is maintained using a back pressure regulator, and this allows the system to be maintained at high pressure. And the resulting product that we would ideally get would be a liquid that looks like this. And it's, as you can see, it's a very clear liquid. It is very pungent as well. But this is a liquid that you would then feed into a hydrogenation reactor to create your fuel product. So a large part of the research that took place here was figuring out what type of catalyst we could use for this process. And by going through previous literature and going through multiple trial and error, we were able to stumble upon a catalyst called zeolite beta, and this is a commercially bought material. However, the novelty in this is that we had to introduce nickel onto the catalyst support itself. So in the first image to the left, we have a impregnation process where we are dripping a nickel nitrate solution into the zeolite beta. The picture in the middle shows the slurry that we will ultimately end up with, and after subsequent drying and calcination steps, we will end up with a grayish powder as shown on the right. So what we're doing here is we're essentially exchanging the nickel onto the material itself using basically an ionic exchange process. So the cations that exist on the support will then be exchanged with the nickel, which produces a nickel zeolite beta type of catalyst. Thankfully, we were able to also do some imaging as well. So we used transmission electron microscopy to take a look at the material at the nanoscale, and these dark spots that are on the that are shown on the image itself represent the formation of nickel oxide clusters, and these are mostly on the external surface of the catalyst. On the internal pores, we have the isolated nickel sites itself. Okay, so now once we've created the catalyst, what's left to do is operate the reactor and determine the effects of different operating parameters on the reaction conditions itself. And as you can see here, in these figures, what we've shown is the selectivity of different olefinic products. We were able to produce butenes all the way up to C10 plus. And the main takeaway from here is that the number of even carbon products is very, very nice. In fact, we end up with very little odd products, and that is very important because the formation of odd carbon products would potentially lead to a inefficient type of catalyst. So this shows that the oligomerization is truly taking place. As you can see, the conversion also increases quite drastically as we increase the reaction pressure. Now, the effect of temperature is also quite important as well, and one that we focused on extensively. One of the main takeaways from the first figure is that at low temperatures, the reaction specifically favors the formation of butenes. For butenes, that's also a very interesting commodity chemical. However, to shift the direction of the oligomerization towards fuel-like products, we will need to increase the temperature. So as we increase the temperature from 30 degrees to 120 degrees, we can see that the butene selectivity drops, whereas the selectivity towards octenes and hexenes, octenes, and so on and so forth increase. So the reaction temperature does play a very large role in this process. Lastly, we wanted to test the ability of the catalyst to maintain its activity over a long period of time. So over the period of 3 days, with some sleep and not much sleep actually, we were able to operate the reactor for 72 hours on stream, and we were able to observe that the activity was able to be maintained at fairly high conversion. There's an initial drop-off in conversion, which is expected. However, what was not expected was that the conversion would be able to maintain itself over long periods of time. So this was quite exciting to see. So the main takeaway of this, of my portion of the project, is that we have found a viable heterogeneous catalyst for ethylene oligomerization. We were able to first and foremost construct a reactor that was capable of accomplishing this reaction. We synthesized an appropriate catalyst for the process, and we were able to do some characterization on it, which is still a work in progress. We've also observed the effects of several reaction operating conditions, such as pressure and temperature on the conversion and the product selectivity. And we also observed that the catalyst can maintain high activity over extended periods of time. Okay. And that concludes my part of the presentation.
Thank you, Chang, Hanna, and Oliver. That was really great, and I appreciate having a chance to hear what everyone has been up to in the conversion lab. Sounds like not getting a lot of sleep. It's the short story. And again, I want to mention that there are a number of undergraduates and also other graduate and postdoc researchers that are involved in advanced hardwood biofuels. And even here at the University of Washington, there are undergraduates getting training, working in the conversion lab here. So at this point, I have a couple of poll questions to start with, if you could answer the polls on the right. And I'm going to read some of the questions that we have in the chat box, and hopefully our speakers will be able to answer them. So first we have a question from Patricia for Chang. Why do you think the sugar yield differs between the low and high productive hybrid poplar?
Okay, so the question is why we have the different sugar yield between low and high productive hybrid poplar. Okay, that's a very good question. So it's more likely to relate to the structure of the lignocellulose. Like the analogy I used before, sugars are locked in the cell wall, but different types of poplar for low and high productive poplar may construct the cell wall differently. Some of them may use the bricks that has more rigid structure, and namely the sugars bound to each other better. And others may have a different type of, may use different types of lignin cement. Say some of the cement holds the bricks better, so during pretreatment or hydrolysis, we cannot totally peel off the individual sugars down. And some of them may be affected at different structure or different fraction of different units of lignin. So those differences can be affected by both the inherent genetic information of the trees as well as the environment the trees were planted. Thank you.
Thanks, Chang. Patricia, let me know if that did not answer your question, but answered it for me. So the next question, also from Patricia, is for Hanna. Are there disadvantages to de-ashing other than costs? And is it correct to say that acid was used in the de-ashing process? So are there reasons not to do the de-ashing process, or what are the downsides to de-ashing?
Yeah, thank you for this question. We lose some sugars because of de-ashing, but then when we do the calculations to get the overall sugar yield, we anyway end up with a better, higher sugar yield for the de-ashed case. So that's apparently not that big a problem. Then the next step is, of course, to design a process and go through the economics, because it's all about the economics. The process cannot take place if it's not economically feasible.
Is there energy involved in the de-ashing process, or how is the de-ashing process working?
We do it at a temperature of 80 degrees Celsius, so but it's not like that high considering a process. Yeah.
So for Oliver, Kat is asking what's the purpose of the nickel in the catalyst?
Oh, that's a great question. So the purpose of the nickel is to basically activate the ethylene. The nickel acts as a kind of like a meeting ground for oligomerization to begin. So what's interesting about this type of catalyst is that there's not just nickel on the support. There's also acidic protons as well. So if you have only the protons in the support, the catalyst will just coke up and not really work out so well. When you have the nickel in addition to the protons, what you have is this initial activation of the ethylene to the material, and then the oligomerization can truly begin at that stage.
Here we have another question for Hanna. What would be the effect of only using hot water leaching?
It have similar patterns, but we won't get as good results with that. But that's also one, like, we have been working on doing that as well as a reference.
So another question for Chang from Patricia. What advice would you give to the feedstock team as they're trying to select the best poplar? What should they be looking for?
Okay, so that's a very interesting question, because if you only stand on the point of feedstock grower, you will only think about the productivity. You will for sure choose the most highest productive clone, the hybrid, to grow. However, when you really sell the product to the biorefinery, if the biorefinery is going to evaluate the quality, say how much sugars can they get from the biomass, the feedstock quality may vary. So currently, as far as I know, there's no such kind of evaluation so far. but it can be a trend in the future. Like by the gate, the biorefinery will evaluate the quality of the coming feedstock. So I feel like it will be good to strengthen the communication between the biorefinery and feedstock grower to find out the optimal choice in terms of the hybrids and the clones. And I think it will both benefit. It will benefit both sides. And so yeah, from that I think the more communication and collaboration will be necessary. Thanks.
So we have a question from Todd Miller. What is understood about the geographic and climate differences to explain the sugar yield differences between the Oregon and California sites? I don't know if this is something that...
Yeah, that's hey Todd, nice to see you here. So actually, we didn't specifically look into the chemical structure based on the effect of different locations, but I can tell you in California side, the collectible side, we experienced a drought like 2 or 3 years ago before the harvest. So I think that's maybe one of the reasons that the environment, the climate did make a significant change to our feedstock and makes the sugar release much difficult, and and also the productivity much lower. But from now, we are still exploring the difference between the sites because of the climate difference.
Great, thanks, Chang. So a question for Oliver from Jeff. What other means beside microscopy did you use to determine the efficiency of adding nickel to the catalyst? And what was the efficiency?
By adding nickel. By efficiency, do you mean the activity of the catalyst? I'm trying to just make sure I understand the question correctly. Oh, this is cool. You can chat back and forth.
If we were even more high tech, we'd have everyone with a microphone, but...
Oh, yeah. That's okay.
Sometimes simpler is better. So I see Jeff is typing. We're waiting to get a clarification of the question.
Nickel state and support. Oh, that's a great question. So what we did is we also use ICP. That's called inductively coupled plasma. I think it's optical emission spectroscopy to determine the amount of nickel that was produced, or the amount of nickel in the original catalyst. And then what we also did was we took the same catalyst that we use in the reactor, and then we also ran ICP on that as well. So I was not able to show those results in this presentation, however, due to time. However, the amount of nickel did not change between the two instances. Not sure.
Jim, can you clarify, is that a question or a statement? Others have reported high effects of nickel content in materials of construction.
Got it.
Great. Thanks, Jim. I had a question for Oliver. You were comparing oligomerization and polymerization, and these aren't words that I use on a daily basis, so you might have to explain the difference
Sure.
again too. When you're talking about oligomerization, lets it stay as a liquid, whereas polymerization would have made it a solid, or can you talk about that again?
So polymers are more like plastics, and those are more like solid materials that we use on a day-to-day basis. However, the difference between oligomerization and polymerization is just the amount of monomeric units that you use. So in polymerization, you would use probably tens of thousands of these ethylene molecules to produce your plastic. However, in oligomerization, we're only taking a very small amount of those ethylene molecules, like say for instance 5 or 6 molecules instead of tens of thousands of them.
And does that make it harder, or is that the reason you need a special catalyst, is to prevent it from getting too big?
Right, because essentially depending on the purpose of what process you're going for, controlling the oligomerization is actually quite difficult, because we had an instance in the lab where we actually polymerized our ethylene and formed a big block of plastic in our reactor. So it was definitely a lot of fine tuning had to go into the process.
Makes a lot of sense. Thanks, Oliver.
No problem.
Do we have other questions from the audience? I'll leave up the chat box for a bit. If you have more questions, type them in, and I'll talk a bit about some of the upcoming events that we have. We're hoping to have a harvest tour in Hayden, Idaho. If you're interested in that tour, please register on our website, and we'll give you updates. There is some question about that, so please do register if you're interested in hearing more. We have 2 webinars coming up. In April, late April, we're going to hear from Greenwood Resources, and they're going to give us an update on what's going on with harvest. They're harvesting Jefferson and hopefully Hayden this year, and they're going to give us a kind of second harvest webinar, round 2 of what's going on now that they have coppice growth coming back. And then in May, we're going to hear about the technical college program that AHB has supported, Walla Walla Community College and Agriculture Center of Excellence for Plant Operations. And we're actually going to hear some of the student success stories and hear from students themselves. So again, you can also register for those 2 events so you can get updates and reminders as we go along. So thank you, everyone. I don't see any more questions coming in, so I think we'll leave it off here. Thank you again to our presenters, Chang Dou, Hanna Horhammer, and Oliver Jan. Have a great day, everyone. And if you want to watch this webinar again or watch our previous webinars, it'll be available on our website later this week. Thank you.